analysis reveal SARS-CoV-2 pathology and cellular targets. So take it away.
Thank you so much for the introduction. Can you see my slides?
Yes.
Great. I'm really excited to talk to you today about how COVID-19 tissue atlases can reveal SARS-CoV-2 pathology and cellular targets. This was a really extensive effort across multiple institutions and hospitals in the Northeast of the US. And I hope you'll be inspired by how our community came together at a really uncertain time to try to learn more about this disease. So our COVID-19 research effort was really centered on creating a tissue biobank from human autopsy donors. And we had three main objectives. The first was to build a cell atlas of infected tissues, describing cell composition and state. And second, we wanted to compare disease versus healthy tissue and ask how cell compositions change during infection. And third, we wanted to assess what cell types tend to be infected and what gene expression programs are turned on in those SARS-CoV-2 infected cells. And in order to answer these questions, we mainly leveraged single cell and spatial RNA sequencing. And both of these technologies provide a high resolution overview of the samples. And we were really fortunate that we were able to quickly engage a network of area hospitals and adapt our existing infrastructure and protocols that we normally use to profile clinical samples to process these SARS-CoV-2 infected tissues. Because the disease has been shown to affect multiple organs, we collected many tissue types that are listed here, including multiple portions of the heart and lung, the lymph nodes, trachea, and spleen. And working with virally infected fresh tissue from deceased donors was new to our lab at the time we started this collection, which was the very beginning of April of last year. Due to this, we processed these samples in a variety of ways, and the goal was to assess which protocol would give us the highest quality data. So the processing protocols that we used include dissociating tissues into single cells as soon as it arrived in the lab, and then profiling these cells immediately. And I should say that the single cell technology that we leveraged for this work was 10x genomics. We also froze biopsy-sized portions of tissue viably, or we flash froze the biopsies for future profiling. And we also preserved larger sections of tissue via FFPE. In the end, we were able to collect samples from 32 donors across 4 hospital sites, and we were able to bank over 420 different tissue specimens. And this is the full sample metadata table that we provide in our paper, but I wanted to zoom in on a subset here just to illustrate some of the properties of the samples that we collected, because I think they're relevant to what I'll talk about next. So again, we collected as many tissue types as we could that are highlighted here, but notably the types of tissue that we were actually able to collect really varied across hospital sites. And each row in this table represents the information from one donor. And the metadata we collected included type of organ failure experienced by the donor, days on ventilation or IMV, days from symptom start to death, and post-mortem interval, which is the time from death to autopsy. And the long PMIs for many of these samples meant that big adjustments were needed in both our experimental and our computational protocols, and able to handle these low quality tissues. And today I'll mainly focus on the results of the lung. So we developed a computational pipeline that we use to tackle these technical challenges that were really unique to this data set. So first, we needed to efficiently and uniformly process a huge amount of data across different tissues. And the goal is to get RNA-seq reads from both human and viral alignments. And to accomplish this, we use the cloud computing platform Terra and the Cumulus pipeline. And then we were also able to align our RNA-seq data to a joint human SARS-CoV-2 reference that we assembled. And a big issue was that we had to deal with ambient RNA contamination. So those long PMIs associated with those samples likely promoted significant cell death and tissue damage. And as a result, we observed really substantial amounts of ambient RNA in our single cell and single nucleus RNA-seq profiles. So to overcome this, we implemented CellBender into our analysis pipeline. So this uses a deep generative model of background contamination counts, and it removes ambient RNA and empty droplets from our data. So here I'm showing cluster U maps, and on these maps, each dot is a cell, and cells with similar gene expression profiles cluster together. Each cluster is a different color, and as you'll see in the next few slides, we can often define the cell types within these clusters. The cells that CellBender actually identified for removal are shown as black dots in the UMAP on the left. And you can see on the right that after CellBender, we really see improved separation of our clusters. Low quality cell removal via CellBender improved our cell type signature detection too. So here I'm showing single nucleus profiles pre and post CellBender, colored by expression of gene sets specific to AT2 cells in the lung. And I hope you can see on the right that post CellBender expression of these genes is more cell type specific. In data that I won't really show today, we actually found that single nucleus RNA sequencing rather than single cell RNA sequencing performed better for these samples. And sort of a take-home message, I think, is that for anyone working with really low quality or diseased tissue, using both single nucleus RNA sequencing and CellBender might be a really good combination. And to provide some context for the lung results that I'm going to talk about today, I just wanted to illustrate one way that the virus can enter cells in the respiratory tract. So on the left, I'm showing a schematic of specialized epithelial cells that I mentioned earlier. They reside in the lung alveoli. They're called alveolar type 2 cells or AT2 cells, and they secrete surfactant. On the right, I'm showing you that SARS-CoV-2 can bind ACE2 on the surface of these AT2 cells, but viral entry actually requires additional cleavage by host proteases like TMPRSS2. So after a really large amount of effort going into optimizing our experimental and computational protocols, we were able to create a COVID-19 lung cell atlas of over 100,000 single cell and single nucleus expression profiles. Again, I'm showing you the cell cluster maps where each dot is a cell, and then cells with similar gene expression profiles cluster together. And now each color on this map represents a different cluster or cell type, which we've labeled here. And to label these clusters, our team developed a novel and automated cell classification approach that leveraged existing annotated single cell data sets of the lung. We also manually annotated the clusters to further refine our cell labels. And with these two approaches, we can see that we were able to sort of recover all of the expected major cell types in the lung. But we also wanted to take a look at the cellular proportions of COVID-19 lungs. And for this analysis, we compared our COVID-19 lung data, again, to other single cell and single nucleus RNA-seq data sets of healthy deceased donors. And here what I'm showing on the right is the mean and 95% confidence intervals of cell proportions in the COVID-19 lung, which are the red bars, or the healthy lung in the blue bars. And the different hues of blue indicate which RNA-seq protocol was used. SC is single cell, SNUC is single nucleus. What we saw was that there is a significant decrease in the proportion of AT2 cells, dropping from 20 to 40% of cells in healthy lung to less than 10% in the COVID-19 lung. And what we think is, because as I showed you earlier, AT2 cells are targets of SARS-CoV-2. So we think this decrease might reflect virally induced AT2 cell death. But we also saw other cell types actually increased in the COVID-19 lung, including fibroblasts, macrophages, myofibroblasts, vascular endothelial cells, and all of this together really suggests a fibrotic and inflamed lung microenvironment in severe COVID-19. We next wanted to identify cell type expression changes associated with severe COVID-19. And to do this, we identified differentially expressed genes between AT2 cells specifically from the COVID lung and the healthy lung. And on this volcano plot, each dot is now a gene. And genes on the right, which are colored in green, are actually upregulated in the COVID-19 lung. And we see within AT2 cells of the COVID lung, there is upregulation of genes associated with the host viral response, inflammation, and the adaptive immune response. And on the left, we see that expression of lung surfactant genes were really reduced. These are shown, reduced genes are shown in left in purple. And this phenotype was previously reported in both in vitro and in vivo. We also saw an increase in the expression of the pre-alveolar type 1 transitional cell state, or PATS program. So this is an intermediate state where AT2 cells can actually transition to AT1 cells during alveolar regeneration. And the PATS program has been shown to be expanded in other lung diseases like pulmonary fibrosis. And this seems to make biological sense because fibrosis has been documented in patients with severe COVID-19. And so the loss of these AT2 cells that I showed you earlier and the expansion of this PATS program in the COVID-19 lung really indicates to us that normal AT2 to AT1 differentiation programs in the lung of COVID-19 patients is compromised. And we have a model of what we think is happening during epithelial cell regeneration. On top, we're showing healthy, where AT2 cells self-renew and differentiate into AT1 cells. And on the bottom, in COVID-19 alveoli, we think that AT2 self-renewal and AT1 differentiation pathways are really inhibited. And this results in that PATS accumulation that I showed you and recruitment of airway-derived intrapulmonary basal-like progenitor cells to the alveoli. Very excitingly, we were able to capture viral RNA in our data. So on the left here, again, I'm just showing you our annotated COVID-19 cell atlas for reference. But next we wanted to see which of these cell types tended to be infected in the lung. And what we found was that most cell types contained some level of virus, but myeloid cells were significantly enriched for viral RNA. And the red color here indicates a positive viral enrichment score. And then notably, the presence of SARS-CoV-2 positive cells actually did not overlap with co-expression of the SARS-CoV-2 entry factors, primarily found in epithelial cells. And we've plotted expression of these factors in the graph on the far right. And so again, we think now that the lack of virally infected epithelial cells in our data is probably due to excessive death of these cells caused by high viral replication and the highly inflammatory lung microenvironment. And I should say that all of the viral levels detected in our RNA-seq data were also verified by viral qPCR of adjacent biopsies that we took from the tissue. And we wanted to gain insight into how COVID-19 affects other tissue types, not just the lung. And so we profiled the heart, liver, and kidney tissues from a subset of our donors. And for this, we use single nucleus RNA sequencing. All of our data for these atlases can be found on the single cell portal. And just to summarize, for the piece of the data that I showed you today, with our improved experimental and computational pipelines, we could successfully build a cell atlas of the COVID-19 lung. We used a novel cell type classification strategy to annotate the lung cells, and then we were able to describe COVID-19 specific lung states like an expanded PATS program. Next, we actually were able to take advantage of existing healthy data sets and look at specific changes that were unique to COVID-19 infection in the lung. And with this, we saw decreased levels of A22 cells and increased levels of immune, endothelial, and fibroblast cells. And our goal going forward will be to further sort of describe how the COVID-19 phenotype varies across tissues, not just the heart, liver, and kidney, but the other tissue types that we collected. And we can also start to figure out what cell types tend to be infected in specific tissues and what gene expression programs are turned on or repressed in these virally infected cells. And data that I didn't show you today, but is in the paper, we found that, again, myeloid cells tend to be infected, but they turn on gene programs involved in cytokine signaling, response to interferon and inflammation. And we have a lot more data in the paper, including spatial RNA sequencing assays and GWAS results, and I hope you'll check it out. So this was a massive, massive effort. I want to thank the entire team who worked really tirelessly during the pandemic to make this work possible. There were many institutional staff that were very critical, working, when everything else was shut down, that allowed us to do this research. And so we thank them. And we really, really give our sincere thanks to the study participants and their families. Thank you so much for the invitation to speak today, and thank you for your time and attention.
All right, I guess, again, we have our question session, and feel free to just speak up or ask through the chat if you'd like us to help ask for you. So I'd like to begin with a question on, I think it was very interesting that the viral RNA was not necessarily detected in cells with the entry proteins. And I think it's definitely really possible that a lot of those cells have died. Can you comment on the cells with obvious viral entry protein expression, but do not have detection? Because it seems like almost all of them did not. Are they just new? Yeah, go ahead.
Yeah, so the majority of the cells with the most sort of entry factors and proteases that you need for the virus to enter are epithelial cells. There are some other cell types that do have those entry factors. And we did detect levels of viral RNA in almost all cell types, but we sort of, we wanted to be really strict with what we defined as infected. And we wanted us, to be able to really robustly detect viral RNA before we called a cell sort of infected is the wrong word, as containing virus. That's another thing we had to watch out for too, because especially with myeloid cells, right? Like it's probably not that they're infected, it's probably that they were eating other dead cells that were actually infected.
Yeah, okay, that makes sense. And are there a subset of myeloid cells that have specifically strong this kind of characteristics?
Macrophages.
Makes sense.
Yeah. Yeah.
There's a question in the chat. Someone asked, did you see COVID-19 infection in lymphocytes?
So we don't, so we started looking at lymph nodes. We're in the piloting stage of that. We didn't look at the blood in this study. And so, yeah, so far we've only looked at lung, liver, heart, and kidney. And then within the lung, primarily the immune cells like macrophages we consider to have high viral content.
Thank you for the great talk. Maybe to follow up one question, but it's really interesting because people, I think, still think that there could be some alternative entry factors in the cell. Could you maybe try to use this data to find out these factors? Also, it's interesting to check whether the stoichiometry of the enrichment of this RNA is somehow different between the cell types, which maybe suggests that there are different life cycle of the virus in different cell types.
Yeah, no, those are both really great points. We haven't done that, but we can try. We did see, and in the cells that we called as like robustly enriched for viral RNA, we did see both viral reads aligning to the positive and negative strand of the virus. So we think that, you know, that could indicate that there was active replication in some of the cells. But that's a really good point.
Might I ask about the spatial configuration, since you guys have done some spatial analysis, and I actually don't know whether in these COVID patients, do they have regions of the lung that were more badly affected than the others, and do you compare those within the same patient?
Yeah, that's a really, really great question. So the first thing we did was we used like an RNA scope assay to try to just look at expression of the virus in the lung. And again, we're looking at deceased, tissue from deceased individuals, very severe COVID-19. And we were really struck by the fact that the virus was not diffuse throughout the lung. It would be that one portion had high amounts of virus, and then in a neighboring portion, almost nothing. And what we were able to do when we used NanoString's GeoMx spatial RNA sequencing technology, there you can actually define regions of interest, and you can sort of tell the machine that you only want to collect probes from those specific regions. So we could pick areas of high and low virus, like in the alveoli, and then compare them. And we actually had a pulmonologist sort of help us try to pick particular regions of the lung that might be of interest. But we really saw that it could vary between patients.
Very interesting. Thank you.
Mm-hmm.
All right, if no one has any more questions, I would like to thank you both for talking and thank everyone for coming, and enjoy the rest of your day.
Thank you so much.
Thank you both. Really appreciate it.